Hi everyone, welcome to this video of cell culture technique. Today we will be explaining two types of cell culture techniques, that is primary and secondary cell culture. So if I move on with the brief background history of cell cultures, mammalian cell cultures that brings back to the 19th century when French physiologist Claude Bernard showed that the animal cells are unable to survive in the water body, but it requires a suitable environment to survive. That this exciting findings found that more reason for the cell culture system. After finishing brief history of the cell culture, now I will move on with the cell culture. And what comes in your mind when you hear the word cell culture? Cell culture means it refers to a removing of cell from an animal or a plant and their subsequent growth in a favorable artificial. And this cell may be removed from the tissue directly or desegregated by the enzymatic or the mechanical means before cultivation. So now let's get into the lab and learn secondary and primary cell culture. Let's go!
Now I will be diving you into the fascinating world of the primary cell culture. Let's start by looking at what is primary cell culture and where it comes from. Primary cell culture is derived directly from the living tissue. When I say living tissue, it can be tissues of plants, animals, or the human. This means that they can retain the characteristics and functions of the original tissues. Now let's look at the examples of the primary cell culture of the plant stem cells. Let's say that we have plants with unique characteristics and we want to study the stem cells of these plants. First, we have to remove this plant's stem cells carefully without harming this plant. After removing the small sections of the plant tissues, you have to isolate the stem cells from these samples. Then the isolated stem cell should be placed into the culture dish containing the nutrient solutions which will optimize the plant cell growth. Over the time, this cell will be multiplied and it will help you to learn the characteristics and functions of the cells. Now let's look at the steps involved in the primary cell culture in the details.
In first step, we have a tissue acquisition. In this, we have to obtain the living tissues from the donor organism, which can be a human, animal, or the plant. Tissue can be obtained through the biopsies, surgical discards, or the other appropriate methods depending on the source. In second, we have a tissue dissections. In this, we have to carefully dissect it to isolate the specific area or the cell type of interest. Then we have a dissociation. In this, the dissected tissue is treated with the enzyme and these individual cells. This step separates the cell from each other and allows them to be cultured independently. In cell isolation, specific cell type of interest are isolated from the dissociated cell mixture. This can be done through the techniques such as filtration, configurations, or the selective adhesions to culture the surface. Culturing cells. In these, cells are incubated under the controlled conditions, typically at the specific temperature and in the humidified atmosphere. The culture medium is replenished periodically to maintain cell health and the viability. In LUS, we have the observations and the maintenance. In these, cells are regularly observed under the microscope to monitor their growth, morphology, and the behavior. These are the steps involved in the primary cell culture.
Now I will be explaining on the advantages and the limitations of the primary cell culture. There are There are many advantages and limitations. However, I will explain only one advantages and one limitations. One advantage of the primary cell culture is the increased relevancy. It means that the primary cells are obtained directly from the living tissues, so it can retain specialized characteristics and the functions of their original states. So this will make more accurate representations. On moving towards the limitations, it is the limited lifespan. It means that they can only undergo a certain number of cell divisions before they die. Therefore, this restricts the amount of time they can be used for the experiments. Therefore, the limited lifespan is the limitations of the primary cell culture.
This is all about primary cell culture. Now I will go over the secondary cell culture. Firstly, the secondary cell culture it is a type of culture of cell that has been derived from the primary cell culture. And there are the main methods for secondary cell cultures. And some of the common methods are subculture, differentiations, and cryopreservations. Firstly, the subculture. This process involves the transfer of cells from one cell culture to other to maintain the cell viability and to promote growth. And second, the differentiation. This differentiation of cells in secondary cell culture involves introducing cells to undergo changes on gene expressions and morphologies to acquire specialized functions or phenotype. And third one is cryopreservation. This method used to preserve cells for long time periods in a freezing ultra-low temperature.
Some of the advantages and limitations of secondary cell cultures. Firstly, the advantages. Increased cell yield. Secondary cell cultures allow for the expansion of cell population, providing larger quantities for experimentations or production. Second advantage are long-term studies. Secondary cultures enable long-term study as cell can be continuously cultured for extended periods. Some of the limitations are firstly the genetic drift. Over time, cells in secondary cultures may undergo genetic changes leading to alterations in their characteristics. And second limitations are technical expertise required. Establishing and maintaining secondary cultures requires specialized skills, equipment, and research which may not be available to all the researchers. So these are some of the advantages and the disadvantages of secondary cell culture.
So I hope you all know what cell culture means and its different types. Now I will be taking you through the applications of cell culture. First one, we have medical research. Cell culture is essential for studying diseases, drug testing, and developing new treatments and therapies. Second, we have drug discovery. So cell culture are used to screen and identify potential drug conditions, speeding up the process of drug development. Third, we have regenerative medicine. Cell culture is crucial in tissue engineering and organ transplantation. Fourth, we have biotechnology. Cell culture is used to produce proteins, vaccines, and antibodies for improving healthcare.
So this is end of our video. I hope you understood what is meant by cell culture and its type of techniques. And our video mainly focuses on brief introduction of cell culture technique, and there is many more to explore. You guys can do it. And thank you and touch the